Hi, hello, welcome to our channels. So now we are going to discussing about types of microscopy. So subject on general microbiology and microbial diversities. Last class we have discussing about our introduction of microbial diversities are discussing detailed discussing about it. So today we will discussing about types of microscopy. So video you have understand all the viewers. I hope to understand in this video. Please subscribe and share it to your friends. So let's start the video. So types of microscope. So types of microscope. First of all, I told you content, history of microscope and principles of microscopy and types of microscope and light microscope and electron microscopic. And last one, summary of types of microscopes. So first of all, I told you what is it microscope? What is it a microscope? So microscopic means being invisible to eye, being invisible to eye unless aided by a microscope. And microscope is a laboratory instrument also. So what is it? Microscope laboratory instrument. It is used to examine objects that are too small to be seen by the naked eye. Okay. So microscopy is the science of investigating small objects and structure. Okay. Next slides also history of microscope. So history of microscope. So many histories interpreted my slides. So particularly important for I told you Robert Hooke in the year 1635. 1635 to 1703. Robert Hooke, he is further he is further refined the compound microscope, aiding such features as a stage to hold the specimens on illuminator. It is used for illuminator and hose and fine focus controls. After that, 1800, the compound microscopes designed by Hooke and others were limited to magnifications of 30x to 50x. Images is largest. And next, authors and history of microscopy for Russian scientist in the year 1933. He built with a scientist name Ruska. So 1933 built the first electron microscope that accessed the resolutions attainable with an optical light microscope. So many history of microscopy. And important I told you. Another one important is Siemens in the year 1939 produced a transmission electron microscopic TEM. In 1939, Siemens produced a transmission electron microscopes. So I give you the detailed history of microscopics. So you see the slides and read and write, and you will take the class notes. Okay, it will be used for lab and practical uses also. So next slides on microscope used by Leeuwenhoek. So microscopic used for Leeuwenhoek. So particularly the diagram of microscope constructed by Anton von Leeuwenhoek in the these videos. Okay. And next few slides on principles of microscopy. So what are the principles? First of all, magnifications. Second one, resolution. And third one, numerical aperture. And fourth, illuminations and apparitions. This is the 5 principles of microscopics. So what is it magnification? Magnification is the factor by which an image appears to be enlarged. It is dependent upon the curvature and size of the lens. So particular images see that power of magnifications. Magnification is particular enlarged images. What is it? The power of magnifications. So already you see that magnification lenses, so many jewelry shop, you see that magnification lenses also. Okay. Next, resolutions. Resolutions are resolving power is ability of a lens to show two adjacent objects as discrete entities. So resolutions means for resolving power. So resolving formula, what are the formulas are present? Wavelength of light in nanometer by 2x numerical aperture of objective lenses. This is also the resolving power formula. And next principles, numerical aperture. So numerical aperture of objective is a measure of it is ability to gather together a light and resolve fine specimen at a fixed objects. So each object has a fixed numerical aperture. Reading higher the numerical aperture, the better will be the resolutions. Greater the refractive index, the greater will be the numerical apertures. You see that picture and right side you see that picture. What are the numerical aperture and the numerical aperture pictures? You see that reproductive index. What are that? And formula is used numerical apertures also. Next slides for illuminations. So illuminations, the effective illumination is required for efficient magnification and resolving power. Artificial light from a tungsten lamp. You see that illuminations, artificial light from a tungsten lamp is the most commonly used light source in microscopic. So in this illuminations, commonly used light source in microscopy. Too much light can reduce contrast and burn out the images. This is the illuminations. Next principle is about apparitions. So apparition is a property of optical systems such as lenses that causes light to be spread out over some region of space rather than focused to a point. Apparitions is associated with microscope or spherical apparition and chromatic apparitions also. So spherical apparitions is a creative curved images. Okay, spherical apparitions it creates a curved image rather than flattened. And next, chromatic apparitions, it will be creates a blurry images like a rainbow. It will be caused by the lenses acting as a prism. Okay. So next discussing about types of microscope. So types of microscope, so light microscopes, what are the types are present? So first one, so many types are present, 4 types, light microscopes and modified microscope and electron microscopes and other microscopes. Okay, so each 4 type of microscope subdivided into the 4 categories are present. First of all, light microscopes. What are the types are present? Bright field microscope, dark field microscope, phase contrast microscope, differential, interference, contrast microscopics. These also the light microscopes are present one. And second one, modified microscopy. So fluorescence microscope, confocal microscope, inverted and comparison and polarized and stereoscopic. These are also the modified microscopies. And third one, electron microscopes. So transmissions electron microscope and scanning electron microscope. This is the third type of microscope. And fourth one, other microscope. What is it, other microscope? What are that are present? First one, digital, scanning probe and acoustic and USB computer. These also the other microscope. So next slides on light microscopy. So discussing next slides, light microscope. So light microscope is an instrument. So light microscope is an instrument. It will be used for visible light and glass lenses to magnify specimens. They are highly accessible and commonly found in schools, colleges and clinical laboratories. It will be used for light microscopes. So you see that picture. The usefulness of any microscope is that it produces better resolutions than the eye. Okay. And next slides, simple microscopy. Simple microscope is a magnifying glass that has a single lens with a short focal length. Okay, simple microscope is a single lens. Next types of microscope, optical microscope. So simple microscope uses a lens or set of lenses to enlarge an object through angular magnifications along with the view and erect enlarged virtual images. So magnifying glasses, loops and eyepiece for telescopes and microscopes for used for optical microscopes. So see the pictures, what are the optical microscope used, telescope. Okay, right side on the pictures you see that. Next slides are compound microscope. So compound microscope, Robert Cook devised the compound microscope and illumination systems in the year 1685. It was one of the and modern compound microscopes. You see the pictures and 2 type of the pictures. You see that modern compound microscopes. You listen here carefully and what are the parts are present in modern compound microscopes. You see that parts of the pictures. Next slides, principles of compound microscopes. So Magnifications from compound microscope occurs in two series of lenses. The objective lens close to the objective to be observed, the first one, and second one ocular lens or eyepiece, one closer to the eye. And third, the objective lens produce on initial magnified real images. So images is again magnified by the ocular lens eyepiece to obtain a final virtual images. Image is received by the eye and converted to a retinal and visual images. And so principles of microscopy is total power of magnifications. So 2000 x, so structure less than 0.2 micrometer cannot be resolved with the compound light microscopes. Next one, bright field microscope, so also known as compound light microscopes. It's also known as compound light. It produces images when light is transmitted through the specimen through differential absorptions and differential refraction. It produces a contrasting images. Produce a dark image against a bright background. Uses on bright field microscope. So it is used for biology, cellular biology and morphological laboratory studies. It can be used to identify basic bacterial cells and parasitic protozoans such as parameciums. So images produced by bright field microscopy, you see that right side. Bone marrow biopsies, you see that right side, the bone marrow biopsies of cynomolgus. A monkey is experimentally infected with hepatitis C virus. See that histological analysis showing the figure bright field microscopics. And next slides, dark field microscope. So specialized type of bright field microscope which has several similarities to the phase contrast microscope. To make a dark field microscope, it is used to visualize living cells which cannot be stained or by destroyed by drying. So it will be used for identification of bacterial cells and visualize the internal organs. And next slides, phase contrast microscopes. This phase contrast microscope produced high contrast images when using a transparent specimens. And next uses, so studying the microbial motility and structure of locomotions such as morphologically characters are identified plant and animal cells and bacterial cells, microbial elements such as bacterial endospores. You see that pictures, images produced by phase contrast microscope. And next slides, differential interference contrast microscope, DIC is also known as, this is the NIC, no mark is microscopic. It is an optical microscope technique. It produces with a detailed view. It uses visualize live and unstained biological samples such as smear from a tissue cultures and waterborne single-celled organisms. It will be used for in this DIC microscopy. This is the diagram for DIC microscopic. You see that pictures, principles of DIC microscopic and images produced by DIC is shown in figure. And next slides, fluorescence microscopic. Specially modified compound microscope uses a mercury arc lamp as a source of UV light. The microscope will also comprise excitation filter. The filter protects the viewer's eye from UV rays. The specimen to be viewed is coated with dyes occurred in fluorescent. This is the minerals which show fluorescence. So specimens to be viewed is coated with dyes, acridine and fluorescence. And next, fluorescence microscopic uses. It will be visualizations of bacterial agents such as Mycobacterium tuberculosis. Identifies the specific antibodies produced against bacterial antigens. And next slides, confocal laser scanning microscopic, CLSM. So confocal laser scanning microscopic. The laser beam used to illuminate the spots on specimens. So you see the picture principle of CLSM used on specimens which are too thick for a light microscope. So it produces 3D and 2D images in computer screen. And uses diagnosing CA cervix, evaluation and diagnosis of basal cells or carcinoma skin cells will be used for CLSM microscope. And polarized microscope. Polarizing microscope is an optical microscope composed of a detector lenses. Two polarized. The polarized is positioned at the light path somewhere between the specimens and one analyzer. Second polarizer. Polarizer transmit one polarization angle of light. And crossed polarizers transmit no light. Okay, what are the uses for polarized microscope? It will be study of fire frightened materials that split a beam of light into two. So amyloidosis for uses. And next, inverted microscope will be used in metallurgy. Double microscope. It produces 2D and 3D images. The uses examinations of cultures in flat bottom dishes. So an examine of parasites. Observation of agglutination in serological. And comparison microscope. To observe two different objects at the same time, it used to compare objectives. Composed of two independent objective lenses joined an optical bridge to a common eyepiece lens. The uses greatly in forensic applications for comparing different traces. And next slides, stereoscopic microscopy. It is a double microscope. It is an optical microscope variant designed observation of a sample and low magnification and low resolutions in this microscope. It provides different angles for each eye to view. The use is forensic application for questionnaire document to examine such as comparison of texture and conditions of paper surfaces, ribbons, pen and pencil points, etc. So you see that pictures, what are the comparison of light microscopy? You see that picture. And next slides. Electron microscope is a technique for obtaining high resolution images of biological and non-biological specimens. Uses a beam of accelerated electrons as a source of illuminations. The wave of electrons are 1 lakh times shorter than waves of visible lights. The electron microscopic images provided the information on structural basis of cell functions and cell disease. And next, scanning electron microscope. SEM produces images by grouping the specimens with a focused electron beam. It does not transmit electrons. It produces streaking three-dimensional realistic images. So, scanning electron microscopic for see the pictures and uses. Next slides on use. SEM can be used in a variety of industrial, commercial and research applications. And scanning electron microscopic are used in material science for research, quality control and poll analysis, and criminal and other forensic investigations utilize scanning electron microscopy, and etc. So many things are used for scanning electron microscopy. You see that picture, images produced by scanning electron microscopy. And next, transmission electron microscopic is uses. What are the differences? High voltage electron beam to illuminate the specimen and create the images. Transmits the electron through the specimens. So transmission electron microscopic, you see that picture. And next one slides, transmission electron uses. It provides topographically, morphological, compositional and crystalline informations for transmission electron microscopic uses. So many uses are used. I told you few points also. You see that slides and read and write so many uses. And next slides, other microscopes. Digital microscope. First one, digital microscope. So, variation of a traditional optical microscope that uses optics and a digital camera to output on images to a monitor, sometimes by means of software running on a computer. So used a wide range of industries such as education, research, medicine, forensics and industrial manufacturing. This is the other microscopes. And next one, other microscope for scanning proof microscope, SPM, that produce an image that represents the structure of surface rather than direct view of surface. So used for nanoscale surface and structures. It included atoms. And next other microscopes, acoustic microscope. It employ very high or ultra high frequency ultrasound. The sound waves produce on enlarged images of a small object, used to visualize image of internal features including defects such as delaminations and voids. This is the used for other microscopic, acoustic microscopic. And next one, USB microscope. So low powered digital microscope which connects to a computer, normally via USB port, used for examining the flat objects like coins, circuit board and banknotes. This is also the USB computer microscope. Next one slides, light microscope and yes, electron microscope. See the pictures. Okay. And next summary of types of microscopes. So microscopes. So you see that magnifications approximately what are the um, so light microscopic approximately the used visible light glass lens up to the 1000x. Condenser that prevents light from directly passing through a specimen. Only light scattered by the specimen is visible. So types of microscopic summaries, so many types, and what are the summaries and description of images, what are there, and typical images is what are there, and special features also I give you there. So I hope this video you could be understand. So thank you viewers. Please be subscribe our channels. So next class I see and we meet you. Thank you viewers. Thank you.